This program is brought to you by Emory University.
Good morning, everybody. This is Monty Donishman. I want to welcome everybody to today's Heart and Vascular Grand Rounds. We have the unique pleasure, and I have the unique pleasure of introducing Dr. Muath Bishawi today, who's going to be giving us an excellent discussion of gene therapy and cardiovascular disease. Title of his talk being Thinking Inside the Box, Gene Therapy and Cardiovascular Disease. Dr. Bishawi joined us this summer after completing his cardiothoracic training at Duke. Um, I've known Dr. Bishawi for the greater part of a decade, and it's really been my honor knowing him over this past 10 years or so. He has joined us in the cardiothoracic faculty, and he has a dual appointment both in cardiothoracic surgery as well as biomedical engineering. His history, he did a BS in biochemistry and a master's in public health. And then he went to medical school at Stony Brook University in New York. We recruited him when I was at Duke from Stony Brook, and he was the first trainee in the I-6 pathway that we recruited about 10 years ago. But he decided that 6 years was not enough time, and so he did a research sabbatical for 3 years, got a PhD at Duke School of Engineering. He was significantly engaged in surgical and engineering research. He has over 100 peer-reviewed manuscripts. He's got many patents. He's started several companies around some of his patents. He has funding from the NIH, from NASA, and from industry. He is really an accomplished researcher, and in fact was one of the only people ever in the history of Duke as a resident to run his own lab and have full PI status because of all the good work that he was doing. Um, he currently has one of his companies fully funded by private equity and is going through the FDA regulatory process to bring one of his inventions to market. And so we were very excited when we recruited him here to Emory, and we're looking forward to all the great things he's going to do. Today's talk is going to be, again, like I said, around gene therapy and some of the exciting work that he's going to be doing here with us, with BME, and with other collaborators. On the clinical side, Muath has hit the ground running. He's been very, very active clinically. His primary interests are adult cardiac surgery and thoracic transplantation, heart and lung transplantation. He's interested in, conventional heart surgery in the high-risk, low EF patient, as well as mechanical circulatory support. And so, um, I look forward to hearing what he has to say. This is going to be a good morning. Thanks, everybody.
Thank you, Manny, for the very kind introduction. Our relationship has been a terrific part of and main reason why I came here to Emory. Really excited to be here today, and I want to tell you a little bit about kind of one of our areas of research around gene therapy. It's an old story, but I think there's been some recent interesting updates, and I would love to hear the discussion afterwards and people's thoughts. So this is very familiar to everyone, sort of the central dogma of biology and what we think of sort of the transcription translation from DNA all the way to proteins. And the reason why I'm showing this is that just to highlight that in cardiovascular medicine, most of the drug therapeutics that we have are really targeting at the protein level. And more recently, we have some therapeutics targeting at mRNA level. But what's been really exciting to me is sort of over the last few years, this kind of innovations within gene editing and genomic engineering, it's often called now, that we can begin to think about therapeutics at the DNA level. And in the cardiovascular system, this is still kind of a new concept. I like to show this slide when I talk about genomic engineering, because to me, the rapid progression of genomic engineering technology, specifically CRISPR, has been really unparalleled, unprecedented. So if you look at just the term CRISPR was only coined 2 decades ago. So it's really not very long ago. And it's only a decade ago, it's just 10 years ago, that there was any success in editing mammalian cells. But today we stand now with multiple CRISPR and gene editing therapies that are clinically approved. So within 10 years of editing just a mammalian cell in cell culture, we have FDA approved therapeutics, and that is really rapid in terms of progression. This is just a recent headline of yet another disease where we have a therapeutic that is targeting the DNA. And this is kind of just a look at some of the trials that are ongoing. You can see many organ systems are covered, and multiple therapies have been approved currently by the FDA. But I want you also to notice that there's really nothing for the heart yet. And I think that should raise a question of why that is, because there's a lot of work that is done on genetic cause of cardiovascular disease, but yet we don't have any therapeutics related to that. So kind of this is where we are today. I want to kind of step back now in time, back to 1998. This is before I even knew what gene therapy was. And to kind of some of the early efforts around gene therapy for the heart here in the United States. These were trials conducted in New York at Cornell by what would be my future PI, Dr. Todd Rosengart, a cardiac surgeon scientist, and his long-term sort of research partner, Ron Crystal, who's a geneticist. And what they've done was they were interested in this idea that we can go to a part of the myocardium that's ischemic, that has no options. So there's no options for revascularization, PCI or CABG. And perhaps we can inject a therapeutic viral vector, adenoviral vector carrying VEGF constructs and try to have those areas overexpress VEGF in an effort to regrow blood vessels. And they coined this sort of sexy term of biological bypass. And there's one main trial with two different cohorts. The first cohort were folks that were going for CABG, but they had areas that did not really have very good targets, and those areas got the VEGF target that's directly injected into the myocardium. And then another group of patients that actually had no revascularization options, and those patients underwent a thoracotomy, and then through that thoracotomy, they had direct injections into the myocardium of the VEGF constructs. That was, 1998. 2008, I was at Stony Brook and doing some research with the cardiac surgeons there. And I remember vividly the day when one of the patients that Dr. Rosengart recognized that he did one of the first injections of VEGF in the US, and that patient was undergoing a cath for new angina, and recognized Dr. Rosengart just walking through the cath lab and called for him. And so we looked at that cath, and Dr. Rosengart was sort of interested in saying, these are some of the areas that we targeted, and we're having a little bit, of tumor blush. Obviously, this is just sort of, we get this small vessel formation in our regular patients with coronary artery disease. But the interest was, I wonder what happened to those patients that got VEGF injection 10 years ago. Was it safe? What are they doing now? And how do they compare to the general population? And that was one of my early forays into clinical research, where we tracked down that group of 30 patients or so, half of which had it as a sole therapy, VEGF as a sole therapy, and the other half in conjunction with CABG. And we showed in this paper, which became actually a really important paper for the FDA, was that long-term, over 10 years, these patients had similar rates of cancer formation, which was the big fear if you're injecting VEGF, and similar rates of other complications as the general population. And their median survival, which again speaks to this, when you have coronary artery disease that severe, was about 10 years. This actually paper was used later to launch a number of randomized controlled trials, which are still ongoing, of testing VEGF therapies, or I guess new vascularization therapies for areas of the heart that are ischemic without revascularization. One of which is Dr. Rosengart's company, which are testing in a placebo-controlled randomized control trial the benefits of doing VEGF injections directly into the myocardium. But this was sort of, I was in during my undergrad years, and then I stayed on in that lab for 2 years after undergrad, and also during medical school, and kind of got to really understand, or at least understand, the problems associated with gene therapy to the heart. And so what I want to propose here is sort of a successful gene therapy strategy for the heart really relies on 4 pillars, and those are the actual gene. You have to go after the right gene. Many diseases have multiple genes involved, and sort of choosing the wrong genes might lead to a sort of failure of that strategy. You have to go after the right disease. Some diseases are just really complicated and have multiple factors involved, and so one gene might not be enough, or a couple of genes might not be enough. You have to use the right vector for whatever it is you need. In those VEGF trials, they use adenovirus, but obviously there are vectors with other unique properties, such as adeno-associated viruses and whatnot. And then finally, which is sort of what I was interested in focusing on, was the delivery approach. I remember, doing a thoracotomy for some of those patients and doing random injections was probably required some new innovations to try to improve that strategy. I'm not showing it here, but obviously one of the big limitations of these random injections, you have expression only in the areas that you inject, and you really can't inject an unlimited number of times. So we needed a better strategy to, for example, if you're going after heart failure, you need expression in the entire myocardium, not just random sort of expressions in different spots. So this is kind of a look at the area of sort of some of the strategies that can be used to deliver a gene therapy to the heart. The most elegant and easiest of which, obviously, is just intracoronary injection. Similar to how you do a left heart cath, you go in with your catheter and inject that down the left main and inject down the right coronary artery. This is a strategy that is the most similar to what we clinically already use. It's easy, it's less invasive than some of the rest. And so right away, this became very popular, but that was a problem, and I'll show you why that is. Other strategy, including giving the viral vector retrograde through the coronary sinus, as shown here. Others include endocardial injections with unique devices that were developed just for injection inside the endocardium. Others have also tried, putting the viral vector inside the pericardium, sort of letting the heart bathe in the viral vector of interest. And then obviously the direct myocardial injection, which was used already in some of those trials. Can't really have a talk about gene therapy for the heart without mentioning the Cupid trial. So many of you may be familiar with this, but the idea here was that SERCA2A was one of the genes involved in calcium handling in the myocardium, the myocardial cells. And so there's sort of a huge background of preclinical research, animal studies demonstrating overexpression of this gene in a heart failure model, increases contractility, increases cardiac output, and improves blood pressure. And so this has gone all the way to this trial that you're seeing here. It's a large-scale, placebo-controlled, randomized control trial. Couple of things that are interesting here. One is, if you notice, they had to screen over 1,500 patients for eligibility to trial, and most of those screening failures were due to neutralizing antibodies. So we about 80% of the general population has neutralizing antibodies to adeno-associated viruses. And so right away, those patients will have to be excluded. So here is sort of an example of using the right vector is critical. And of those screened, about 250 of which were randomized, about 120 in each arm were randomized to either undergoing an intracoronary sort of injection of this SERCA2A versus a placebo. So really nicely well-designed trial with a high level sort of evidence, but it completely failed. There was absolutely no difference between the groups. There's not even a signal to go after, despite the fact that we had a large body of literature and preclinical data to demonstrate, to at least suggest that there is a clinical benefit of overexpression in this gene. So why is that? And it wasn't really till a couple of years afterwards, till some of these patients, these are heart failure patients, some of them underwent LVAD and some of them underwent transplant, and that access to some of those hearts that had the therapy given was available. And right away, it could be very obvious why this trial failed. One is that when you're looking at the viral vector uptake, so when you look at sort of specific vector DNA per microgram of DNA that's extracted, it was very, very low. So it's 20 to 500 copies compared to thousands and thousands of copies in the preclinical studies. The other interesting part to me was that less than 1 cardiomyocytes had any evidence of viral vector delivery. So I think right away it became obvious that the issue here was we didn't even test if the gene was therapeutic or not. We just could not even deliver the gene to the myocardium. And so I think this was really a failure of the delivery technique. And the problem here is that it's a one pass through the coronaries into the coronary sinus, and then sort of it gets ejected out. And once it's ejected out, it's obviously filtered by the liver, and then the therapy itself becomes ineffective. So I think after the Cupid trial and some other trials similarly using the same delivery technique, it was obvious that one of the most important things that needs to be developed for cardiac gene therapy is a better delivery approach and a better delivery technique. This one has to have targeted delivery to the heart, has to have multiple passes, You want less secondary organ exposure. It has to obviously be safe, and then with a high transduction efficiency. And it also has to fit within the clinical workflow that we have today. There has been other techniques that were developed that are sort of very different than the regular clinical workflow, and they really never gained popularity because of that. There was a time when I was joining Duke that there was this rise of these machines, these perfusion machines for organ transplantation. Obviously, my background in gene therapy and my interest in transplant sort of, sort of unique and sort of parallel fields that were developing at the same time. And when I saw these perfusion machines, the obvious question was, can they be used for viral vector to delivery to hearts? And there are unique advantages of these machines. The heart is placed on this machine. It is a closed loop circuit. There is no liver in the circuit to suck up your viral vectors. But more importantly, it was warm temperature. And we know that temperature is important for transfection efficiency. And so it was kind of an interesting concept, and we wanted to test it out. Obviously, these machines were developed not for therapeutic delivery to the heart, but they were developed to prolong the time the heart can be outside the body, and also to open up this new area of DCD donors. And so, for example, during my time at Duke, we did the first DCD for adults and the first DCD for pediatric population, and using these machines. And right now, over 300 of those hearts were transplanted using these machines. So they certainly opened up a new area, new donors that can be used. But from a research standpoint, the question was, can these machines be used for viral vector delivery to the heart and kind of overcome some of the challenges that were previously seen with other methods of gene delivery to the heart? And so to do this, we wanted to really use the clinically available machine to make this very clinically translatable. And we had to develop a large animal model of heart transplantation. So as seen here, this is our model where we take donor hearts, we place them on this machine for a 2-hour perfusion run. Again, very similar to what we clinically do. Obviously, clinically, we probably perfuse a little bit longer than that. And then we take the heart out of the circuit, and we implant it in the abdomen of a recipient animal. And so this method of heterotopic heart transplantation allows us to study the allograft, to look at gene expression in the allograft without while the recipient is still alive. So even if the allograft rejects or there's issues with the allograft, we can still get the heart out and kind of sort of look into what we're interested in. And so the anastomoses are the donor aorta to the abdominal aorta infrarenally, and the donor PA to the abdominal IVC. And I've done some of these transplants with Mani when he was at Duke. He would come into lab, and after doing clinical transplants, and him and I would do these animal transplants, and it was really a lot of fun. And here's a look at what sort of the experimental approach is. So this is the donor heart. We then take donor blood from the donor pig, and we actually wash the blood using a cell saver device, and I'll show you why that is. And then we give the viral vector of interest while it's on a circuit, and then we can actually sample the circuit blood over time to look at transfection efficiency of the virus. So we can know very quickly if there's an issue. One of the big questions early on was that if we give the virus on the circuit, will it stick to the tubing? Will it stick to the oxygenator? Will it still be effective? And so we really had to figure out a lot of that out and sort of modify the circuit in a few manner to try to allow us to do this viral vector. These are some of the early kind of studies of feasibility that we did, looking at, the OCS device, this ex vivo perfusion device, has a proprietary solution that you have to mix with the donor blood. And so one of the big questions was there anything in this solution that would inhibit viral vector cell induction? And so we developed these cell-based assays, screening assays that allow us to kind of quickly detect if there's an issue. And the actual proprietary solution had no issues. But when we mixed it with the donor blood, as you see on figure B, there's a significant decrease in transfection efficiency. And that was really due to these neutralizing antibodies that were in the blood. And so once we wash the blood, we reconstitute the blood, and then we're able to re-achieve good transfection efficiency. And when we place the heart in figure D, you see here, we can monitor transfection efficiency over time. We can sort of sample the blood that's circulating, and we can make sure that it continues to be active. And here, up to 2 hours, we had great activity of our viral vector. This is what it looks like. Here's a pig heart on a device, and then to the right, it's very unnatural sight of seeing a beating heart next to the colon. This is the colon right here. I made a lot of jokes with our research team if maybe we can develop sort of colonoscopy-based methods to TEE the heart. But nonetheless, we can actually, because we're just closing the abdominal wall on top of the heart, you can palpate the heart, you can do a trans-abdominal echocardiogram of the heart, you can monitor function that way. This is obviously an unloaded heart. However, it's perfusing, it's beating, and then as the heart starts to reject, we can see it starts with sort of rhythm abnormalities that we see. We can biopsy and sample the heart and look at our genes of interest that way. I think this is probably the most exciting data I'll show is that when we do this method, we have unparalleled transfection efficiency. We can get our therapeutic gene of interest to really the entire heart. So both the right ventricle and the left ventricle, these are thousands and thousands of folds of expression. We did activity assays, luciferase activity assays. So in here, we used the firefly luciferase gene, sort of a reporter gene, to try to figure out exactly where the viral vector is going and what's getting transfected. And we see here that everything is getting transfected. So whether it's the coronaries, the septum, the atria, the ventricles, we really can get it everywhere. And that was, I think, critical when we start to think about what diseases we want to go after. So for example, if we're going after heart failure, we really want to try to get it to the entire myocardium. If we're going after CAV, we want to make sure that it's getting into the coronaries and so on and so forth. And this is kind of a look at sort of, we have this really beautiful pattern of homogeneous expression of a viral vector. So here's one of our reporter genes. Look at the cross section of a coronary, we get it all throughout the coronary, and look at the myocardium, we sort of get expression in the entire myocardium, F as compared to E, and then for the coronary, D as compared to C. And also equally as important, we don't get any bleed through the virus to other organs. So after we do the implant, so after we put on the device, we re-arrest it, we do the implant, the virus is not sort of leaving and going to other areas. And I think that's important from a safety standpoint. You know, people talk about Jesse Gilfinger's case many decades ago as sort of an important safety milestone that has really halted the progression of the gene therapy world for a while. And people say, oh, well, you know, we're using AAV now, it's not a problem. Well, guess what? Last year, there was an AAV-associated mortality, and it was really an immune mortality against the vector capsid itself. So it still does happen. And so targeted delivery to the organ of interest while minimizing exposure to other organs, I think, is still a critical problem that we need to go after. So here, again, we have beautiful expression in the allograft and no detectable expression, even with the most sensitive methods to look at expression in anywhere else. We also further developed this method outside of adenoviruses. So we looked at adeno-associated viruses. There's another paper from the lab that we kind of looked at adeno-associated viruses and started kind of thinking about dose escalation therapies, adeno-associated viruses. In clinical trials today, for humans, for adults, the sort of titration is 1 to 10 to the 14th. So we can actually go much lower than that titration and still achieve really good transfection efficiency. And again, it's only the heart being exposed here, not the rest of the body. Working with the interventional cardiology colleagues, we also developed some methods to, you know, we used to just do a small laparotomy and biopsy the heart that way and sort of look for expression, but we also developed new methods where we can go from the femoral vein of the pig with a biotome and actually still biopsy and sample the heart, similar to how you would for a heart transplant, but this one is going through the femoral vein into the IVC, and then we can turn around and take a biopsy of the RV and kind of look at expression and rejection that way. So then, you know, okay, we developed this method to deliver to the heart. So what gene to go after? And I think that is kind of one of the areas of active investigation. It's a really difficult problem because there's really not that many diseases where one gene can really turn around the phenotype of the disease. But there's a lot of areas within that to think about. So from a transplant standpoint, you know, obviously the three things we think a lot about are primary graft dysfunction, which, to be honest with you, I don't think a gene therapy strategy is that effective, because when we see primary graft dysfunction, it's very early on, and it takes some of these viral vectors a few days to begin to express. So adeno-associated viruses will take almost a week to 2 weeks to begin to have good expression. Adenoviruses, a couple of days. But nonetheless, the primary graft dysfunction timeline is probably happening a little bit earlier. But there are other pharmaceutical therapies and other quick to expression therapies that we can think about there. The obvious problem is rejection, and then finally CAV. And so these are the big areas, at least to think about. And then for coronary artery disease, there's been a lot of investigation around a number of different things, all of which are being tested in some type of clinical trial or some type of preclinical model. But to me, it's obviously, what's really interesting to me is from a transplant standpoint is that, you know, it's a difficult problem. It's not really just one gene. There are multiple redundant pathways in transplant immunology. So if you sort of intervene on one gene, you can just have other pathways that get activated. But we do have a number of interesting things to go after in transplant. So, you know, oftentimes people think about tolerance. We want just complete tolerance of the transplanted organ, but I do think there's still some value of simply decreasing the immunogenicity of an organ. So can we transplant organs with less immunosuppression? Because I think if we can do that, we can really address a lot of the problems we see in organ transplantations today. So most of the problems are obviously either organ rejection or some side effect of the immunosuppression. So whether it's infection, whether it's secondary organ effects, or whether it's malignancy formation. And all those things are happening at the same time, you're still seeing rejection. So I think the ability to decrease the amount of immunosuppression we can give to immunosuppress for a transplant, I think is still be a big success. So one of the genes we started looking into was actually interesting. It's kind of taking a page out of cancer's playbook. Is, can we overexpress PD-L1 as a strategy to downregulate activated T cells? So kind of one gene that may have an effect in sort of cell-mediated rejection. But sort of we thought about this strategy, but what was interesting to me is that we actually didn't know if the heart expresses PD-L1. And we knew from some of the cancer sort of therapy trials, there are some reactions in the heart, but it's really unclear, is it the cardiomyocytes or is it T cells in the heart that could be a problem? So one of the first things we did was to actually look at hearts in our biobank that had rejection versus donor hearts that were controls that did not. And very quickly, we can notice that hearts that had CAV actually had low levels of PD-L1 expression. So I think there may be a role of overexpressing PD-L1 in the allograft as a method to maybe decrease some of the allograft rejection that we're seeing. But that's sort of simply an overexpression strategy. But there are actually very interesting other developments in the world of gene therapy that I think we can think about. So this is a paper that we put together, a big collaborative group between our group and some other groups across cell biology. And what this is a lab, Ken Pass's lab. What they developed is sort of they work a lot on zebrafish and regeneration in zebrafish. And what they've isolated and cloned is this tissue regeneration enhancer element. They call them trees. And the idea here is that if you take a zebrafish heart and you take part of the heart out, that zebrafish can regenerate the heart. And so they were able to isolate these enhancer elements that are activated after injury to the heart. And they were able to then clone some of those in mice. And what we were interested in is, okay, well, if we do that, can we then get a gene to turn on only after an injury or after an insult and then turn back off when the insult is gone? And so we cloned some of these tree vectors into mice and pigs. And then we would give the tree vector that sort of expressing a reporter gene initially, 2 weeks before any type of injury. And we could look and see if there's any activation of our reporter gene, and we don't see anything. And then after we create a myocardial infarction in those animals, the tree vector turns on and turns on our reporter gene. So kind of a very nice system of genomic engineering that you can turn on a gene only at the time of injury. So currently I'll continue my collaborations with Ken Pass to try to continue to develop this idea of a turn on and off switch after injury for the heart as a part of a gene therapy strategy. The other thing we're working on currently is, okay, well, how about if we go after the MSC system? Obviously, it's a very complex and involved system, but there's been interesting developments where you can use CRISPR to knock out beta-2M, which is an important protein subunit for the overall assembly of the MSC1. And then once you knock it out, you actually don't get proper assembly of MSC1. So if you do that, can you make an organ sort of lacking or cells that are lacking MSC1? So interestingly, I had this idea and we submitted a grant about this. And the main problem with the grant from a reviewer standpoint was that they didn't think this was feasible. But just a couple of years after that, multiple studies have been published demonstrating the feasibility of this in stem cells. So now we have stem cells that are completely lacking MSC1. Now they have to overexpress a self-antigen. So in some of these data, they overexpress HLA-E, so to avoid killing by NK cells. But nonetheless, it was still a viable strategy to make these cells sort of unrecognizable by the immune system. So we're using a very similar strategy for whole organ editing that way. And as you can see here, there's more genes and more things to test. And so it becomes really difficult to do this in a pig model, right? It's to cause a lot of virus. It's much more complex. Scheduling these search much more complex. So to have a screening system, we kind of developed a number of screening systems. The first is a mouse heart transplant model. is obviously a workhorse in transplant immunology. But in these models, so here's some of our papers looking at this mouse heart transplant model, looking at the effect of different things, including extracellular mitochondria. But then I sort of redesigned the ex vivo perfusion system to a mouse ex vivo perfusion system, which significantly decreases circulating volume of that system from 1.5 liters down to 50 cc's. And now we could use the system for rapid screening of genes, but also it opens a whole new world of the mouse genetics. So obviously we have knockout mice strains that we can then use as positive and negative controls that make it much easier to study the effect of our ex vivo perfusion strategy and gene editing strategy. And so once we take the donor heart, we put it on this machine and then implant the heart either in the abdomen or the neck, and then kind of look at the effect. Other screening models that we developed, this is my old PhD lab. We developed these tissue engineered blood vessels where we can take endothelial cells and smooth muscle cells and formulate these small blood vessels. They are vasoreactive. They have a lumen. We can sort of introduce physiologic shear stress and physiologic flow conditions to these. But this is important, for example, studying things like CAV, where we have a very similar model, at least a more similar model than that of mouse model. So we can take actually human endothelial cells and seed these in these tissue engineered blood vessels, and we can study them that way. Other screening models involves collaboration with the Bursec lab that we looked at these cardiac patches. These are either rat cardiomyocytes or human iPS derived cardiomyocytes. And these patches are, you see here, they're contractile. We can look at their contractility. We can look at action potential duration. We can look at forces of contractility and so on and so forth. So as we sort of continue to screen through some of the genes of interest, we can again take human cells, develop these, and screen our therapeutics that way for rapid screening to try to figure out what genes may be worthwhile to go after in larger studies. So this is kind of just a little bit of an overview of some of the work that we are doing here at Emory. This is my overall kind of interest from a clinical research standpoint, interested in kind of variability in a lot of our practice patterns around transplant, around cardiac surgery. It's actually very interesting to see how much variability there exists nationally and how can we address that variability as a method to continue to improve outcomes. Obviously, I mentioned a little bit about some of our work with ex vivo function and assessment of modifying organs, both heart and lungs. And then using a lot of that BME background to come up with interesting kind of models to use to screen genes of interest. And then I didn't talk much about it here, but device development, design, and spinning out technologies that can make it to clinical practice. And we have a couple of technologies, including technologies around this ex vivo gene perfusion. I think there's more and more interest in gene perfusion. The big pharma companies are getting in the game. Their M&A activity is becoming more and more active around gene therapeutics. So I think we'll be seeing more and more of that. And I think one thing here to consider is kind of also an interesting area is that how do you price gene therapy? So imagine, for example, for hypertension, let's say someone comes up with a therapeutic that cures hypertension. Okay, how do you price that? And so our entire health system is not set up to price gene therapies. Can you give a therapy that costs $1 million but completely cures a disease? Even if we have access to these kind of therapies, we can't really use them yet because our health system is not set up to address them. So I think there's going to be a lot of interesting discussions and problems that we would have to solve as these therapies make it to market. As you could probably tell, I was very, very fortunate during my career so far, and kind of the amount of mentorship I've gotten in the lab and clinically, and how sort of I was able to work on really interesting problems in the lab and get really phenomenal, unparalleled training clinically. And that started as an undergraduate, after undergrad, medical school, and then during my 10 years at Duke. And that is through both our clinical enterprise, but also through the engineering enterprise. Seen here are some of my mentors clinically and research-wise, including my lab back at Duke. But I've only been here a few months, and right away I've started making some connections and friends, both in the research world. You see here some of the cardiology fellows that are in the Joe lab. Dr. Joe has been very kind in sort of helping me get started. Some of the folks in the transplant center, obviously a lot of our surgeons here at Emory, been really terrific partners and mentors. And And I've interacted a lot with some of the folks from cardiology, whether it's intervention cardiology or heart failure cardiology, as we collaborate more and more on patients, and really learning a ton from you guys. And sort of it's exactly the environment I was hoping to find here, which is the environment I came from, this incredibly collaborative environment, both in the lab and clinically, to try to really deliver cutting edge, cutting edge medicine to patients, but also continuing to push the envelope in the lab. And then I'm starting as I start my own lab here, I made my first hire. Here's Jungwoo Kim, and then making a couple more hires, and hopefully the lab will attract some interest from some postdocs interested in this idea of gene modification for whole organ perfusion. I'll end here. I'll say I'm open to business, both clinically and for research collaboration. This is my contact information, and I'll be happy to take any questions or have any discussions. Thank you.
Hey, Moat, that was a fantastic talk. And it's a really great primer on gene therapy, and kind of like how we've evolved. You've done some really wonderful work. Especially I think the things that excite me personally are the potentials for providing gene therapy on mechanical circulatory support to these organs. I think one of the things you touched upon was potentially decreasing immunosuppression. The other aspects of it is potentially mitigating things like primary graft dysfunction and things like that. What thoughts do you have around, do you think that there is some, easy to target, you know, pathway around PGD, or, what are your thoughts on utilizing gene therapy for PGD prevention?
Yeah, it's a great question, Mani. I think so. I think there's two. I think to go after initial PGD. So the PGD we see when we take the cross clamp off and the heart, starts beating, and then within like half an hour, as you're starting to come off bypass, the heart sort of stops beating as well. And I think that early, I think, is difficult from at least a gene therapy. You can give therapeutics, so there's a lot of potential, I think, of giving reperfusion injury drugs, sort of scavengers, damage associated molecular pattern scavengers, and so on. I think what's interesting, though, from a gene therapy standpoint, is I think that early PGD is important for the long-term outcome of an organ. So I think, organs that have early PGDs are those also the more likely to develop CAV long-term. So if we are seeing kind of an early signal, does that translate to a delayed issue with the organ long term, that even if we get them through the early sort of PGD? And so that's where I think I have a lot of interest. So our mouse model, we have actually a CAV mouse model, and part of that model is the ischemic time an organ, sort of the ischemic hit the organ takes early on. So I do think there's going to be some potential of sort of whether it's endothelial dysfunction long term that maybe accelerates CAV formation, smooth muscle cell hyperplasia that might also play a role. Very similar, I think, ideas to what we think about when we think of just normal coronary artery disease, but there's, I think, unique considerations in transplant. The other area of interest is sort of, can you have an organ that maybe experiences PGD? Again, similarly, these tree vectors that I mentioned that we're working on is, if an organ experiences PGD, can it turn on a gene of interest to try to get through that? Whether it's a gene to improve, for example, calcium handling in the heart, to maybe try to decrease the amount of time it requires mechanical support. I think these are all active areas in investigation. I think what's exciting about this is it's totally fertile. This field is totally fertile. There's really not a lot of there's a lot of questions to be answered, including whether or not giving a viral vector to a transplanted organ and having the organ display caspid proteins on its cell surface, is that harmful from a transplantation standpoint? And we don't know. So these are a lot of the questions that we're trying to address.
Yeah, I think that's exciting stuff. One of the first mechanisms, as around this was done in lung transplant. Where the Toronto group used adenoviral vectors to increase IL-10, I think, production in donor lungs, and they actually found probably worse graft function. What are your thoughts around that, around viral vector choice? I think some people argue that maybe it wasn't the IL-10 that was the issue, but it was the fact that they used adenovirus. What are your thoughts around the relevance of which of the vectors that are using, and is viral vectors the best method? Are there other gene delivery methods that you think would make more sense? Is there a difference between heart and lung? What are your thoughts?
Yeah, I think vector choice is critical. It really depends on what you're trying to do. So if you want to express, let's say, a protein for a short period of time versus something that you want to express for years, obviously that changes your viral vector choice. Where you want to deliver it matters. I think there's a whole area of research, and there are big labs just dedicated on caspid design. They're designing interesting caspids that target organs that way. So you can think about, there's a group that works on you know, designing adeno-associated viral caspits that evade the liver. So the idea here is that their bioavailability is much longer because they're not being uptaken by hepatocytes. So I think there's some interest there now. And then there are other ways to deliver your gene of interest, or let's just say a CRISPR construct to a cell of interest. And there are people that have worked on these carbon dots that you can deliver that way, lipid particles you can deliver that way. There's a lot of, I think, interesting approaches. I mean, as you could probably tell from our lab, we're really not married to a certain virus. We're not married to a certain delivery strategy. We're not married to a certain gene. We're really interested in the problem. And, sort of we'll develop the strategies that we need to develop to sort of address the problem, and to make sure we just have, all we care about is how can we get the genetic information we want to the cell that we want. And everything else outside of that is sort of open to changes and innovation and movement. I think that's sort of the nice advantage of being a newer lab, is that you're less sort of your sunk cost fallacy doesn't exist. I'm not married to this one protein I've been studying for 15 years, but rather I'm interested in kind of seeing where how the field changes. So like when the tree stuff came up, it was very easy for us to quickly collaborate on that project and sort of try to figure out, okay, can we use this on off switch for a gene of interest, and so on. The Toronto group has been really inspirational group for me. I mean, they're using, they're working on the lungs, which I think is a much more difficult organ than the heart. Obviously, we have interest in the lung, but I think a lot of the early focus will be on the heart because of all the problems associated with the lungs, as the lungs are constantly being exposed to new, to new proteins that make them immunogenic.
Well, thank you.
I have a question.
Anyone anyone else have a question? Yeah, go ahead, please.
Yeah. Thank you for a great talk. I have a question, about sort of collateral formation, for example. And, we say like people who exercise a lot, it's the repetitive, activity over time that leads to formation. So I'm just wondering, with all of these techniques, like the VEGF thing, once it's delivered to the myocardium, are there techniques to kind of stimulate or boost, just to kind of make that effective? Because I'm just thinking like one-time injection versus repetitive stimulus.
Yeah, you can, it's a great question. I think you can use a different virus. So if you use AAV, you can have long-term expression, years, sometimes, sometimes a decade. If you use adenovirus, it's shorter. It could be a few weeks. The challenge you have with using a viral vector for delivery is that you're limited to how many times you can give that vector. So you develop antibodies against the vector caspid after the first or second injection. So it becomes more difficult to give, like, let's say, the vector at multiple time points. But rather there is where Tumani's sort of point is the vector choice matters a lot. So if you want expression for a long period of time, you want to give something that lasts a long time. For VEGF, I think it's worrisome to give, for example, VEGF with the adeno-associated virus, because you really don't want VEGF expression, high levels of VEGF expression for years. Because this is, again, a lot of the fear around, sort of, for example, cancer formation that way. So yes, you can do it. You can do it based on viral vector. Now, the other strategy you can use is some of these genomic engineering strategies that I mentioned, where you have, let's say, there is an ischemic insult, and only when there is an ischemic insult does the gene express, and then it turns off, which is critical. It turns off when that insult is gone. So similar to I think how our biology behaves, right? You are responding to some type of stimuli, and then when that stimuli is gone, you stop. And so I think that is a critical sort of system on-off system that I think will have an important role in how we think about these strategies in the future.
Okay, great. Well, we'll close. And thank you, Mani, also for leading the discussion. Thank you for an amazing talk. And welcome to Emory.
Thanks all very much.
Pooja.
Thanks for
Thanks,
having me.
everybody.
Mhm.
Nice job,
Thank you.
Mark. Thanks.
Thank you.
The preceding program is copyrighted by Emory University.